Welcome back, everybody. Today, we're going deep on something that could, really change the world as we know it.
Yeah.
Gene drive. It's
Mhm.
kind of like a chain reaction, but with genes.
Right.
A particular gene can spread through a whole population, even if it's not good for the organism itself.
Yeah, that's the crazy part.
So you might be thinking, why would we want to do that?
Right. Right.
Well, the potential is huge. I mean, we could control invasive species,
Mm.
maybe even wipe out diseases like malaria.
That's right.
But obviously, that kind of power comes with a lot of questions,
Absolutely.
especially about what might happen that we don't expect.
Yeah, like unintended consequences.
Exactly.
Like we're messing with evolution itself. It's
Yeah.
exciting and scary at the same time.
For sure. And to really understand this complex topic,
Yeah.
we're diving into some brand new research on a specific type of gene drive.
Boy.
It's called Cleave and Rescue, or CLVR for short.
CLVR. Okay.
We've got these scientific papers. Some are about CLVR in animals, and others, surprisingly, are in plants.
It's wild how adaptable this technology is.
I know, right?
Yeah.
So our mission today is to break down CLVR,
Okay.
see how it works, and explore what it could do in different fields.
Sounds good.
We'll also talk about using it responsibly
Right.
and the challenges of controlling something this powerful.
Definitely important to consider those things.
For sure.
So CLVR works on this really interesting principle called the toxin antidote concept.
Toxin antidote. Okay.
It's like, imagine a hostage situation.
Okay.
The hostage taker, that's the toxin,
Uh-huh.
they demand a specific ransom to let the hostages go.
Okay.
That ransom is the antidote.
I'm following you.
Now, in this case, the ransom is the gene drive itself.
Okay.
Only those who inherit it survive.
Wow, that's a pretty intense analogy.
Yeah.
Survival of the fittest,
Right?
but with a human-engineered twist.
Exactly.
It seems almost too clever to be true.
Uh-huh.
So how does CLVR actually make this happen at a molecular level?
So it uses CRISPR-Cas9. That's like a pair of molecular scissors.
Oh, I see.
CLVR uses CRISPR to target and cut an essential gene in an organism.
Mm-hmm.
But here's the really clever part.
Yeah.
It also provides a rescue copy of that gene.
Okay.
But this copy is immune to those molecular scissors.
So it's like replacing a vital part in a machine
Right.
with a version that can't be taken out.
Exactly.
That's fascinating. But what makes CLVR different from other gene drives?
Well, one key difference is its potential for control.
Okay.
Unlike some that could spread forever,
Uh-huh.
CLVR can be designed to be self-limiting.
Self-limiting. So it eventually stops spreading.
Yes, exactly.
That's a big deal, right?
It is. It's a really important safety feature.
Mm-hmm.
It helps address those concerns about unintended ecological consequences.
Okay, so that's huge.
Yeah,
But how do you actually create a self-limiting gene drive?
right.
Is there an off switch built into CLVR?
One way is using something called a daisy field gene drive.
A daisy field gene drive.
Yeah, imagine a field of daisies. Each one represents a part of the gene drive.
Mm-hmm.
As the drive spreads, this daisy field gets diluted,
Mm-hmm.
and eventually it stops spreading.
Okay, so it's like the gene drive is running out of steam.
Exactly.
That's a really cool idea.
That's a pretty elegant solution.
Yeah, but wouldn't that make the initial spread less effective?
That's a good point.
It seems like a trade-off between control and effectiveness.
You're right. It is a balancing act.
Mm.
But scientists are looking at strategies like supplementation.
Okay.
They periodically introduce new individuals with the complete daisy field.
Oh, I see.
This helps maintain the drive's effectiveness while still keeping it under control.
So you're basically giving the gene drive a boost every now and then.
Yeah, exactly.
Pretty clever.
It's a work in progress.
Okay, so we've talked about control.
Right.
But what about the impact?
Ah.
Where could CLVR really make a difference?
One area with huge potential is controlling invasive species.
Okay.
Imagine a world without the damage caused by, say, gray squirrels. They've wiped out native populations in some areas.
Yeah.
CLVR could manage their numbers
Mm-hmm.
and restore balance to the ecosystem.
That's a great example.
Yeah.
But what about the ethics of basically engineering a species out of existence?
It's definitely something to think about.
Yeah.
Using gene drive technology, especially for that purpose,
Uh-huh.
it raises ethical questions about our role in nature.
Right.
We need to have open discussions,
Yeah.
weigh the benefits against the risks,
Yeah.
and be really careful how we proceed.
For sure. And speaking of benefits,
Yeah,
I'm really interested in CLVR's potential for disease control.
that's a big one.
This is where things could really change the game.
Absolutely. There's research going on right now
Oh, wow.
to use CLVR to control disease vectors,
Like,
mosquitoes that carry malaria.
right.
Getting rid of these devastating diseases would be huge.
Yeah, it could save so many lives.
Exactly.
But again, we need to be really careful about the ecological
You're
consequences.
absolutely right.
It's kind of a paradox.
Yeah.
A technology that could save millions of lives,
Mm-hmm.
but also carries the risk of harming entire ecosystems.
It's a lot of responsibility for the scientists working on this.
I bet it is.
They are very aware of the ethical considerations,
Mm-hmm.
and they're working hard to develop safeguards
Okay.
and be transparent in their research and how they use it.
That's good to hear.
Yeah.
So we've covered the toxin antidote mechanism.
Right.
The idea of self-limiting gene drives like the daisy field approach,
Mm-hmm.
and some potential applications in conservation and disease control.
Yeah.
But I'm curious, what does CLVR actually look like in action?
So there's some really interesting research with fruit flies.
Okay.
Scientists use CLVR to target a gene they need to survive,
Hmm.
and they were able to drastically reduce the fly population in a controlled environment.
So they've shown it works in a lab setting,
Exactly.
but using it in the real world for invasive species or disease vectors,
Right.
that must be a whole other challenge.
It is definitely a big step.
Yeah.
There are a lot of hurdles to overcome.
Like,
Understanding complex ecological interactions,
mm-hmm.
addressing public concerns,
Yeah.
and making sure we use it responsibly.
Right. And speaking of real-world scenarios, we mentioned that CLVR isn't just for animals.
That's right.
It's also being explored in plants,
Uh-huh.
which opens up a whole new set of possibilities and questions.
Exactly. And that's where things get really interesting.
Okay.
We'll dive into CLVR in plants,
Okay.
explore how it could be used in agriculture,
Mm-hmm.
and consider how it might change the future of food production.
I'm hooked. I can't wait to hear more about CLVR in plants
It's pretty amazing stuff.
and what it could mean for our food.
Yeah, it has the potential to change everything.
Yeah.
So using CLVR in plants is fascinating
Yeah.
because there are so many potential applications.
Like what?
Well, we could engineer resistance to pests or diseases,
Hmm.
reduce our reliance on chemical pesticides,
Oh, okay.
and really boost food security.
So we could grow crops that are like naturally resistant.
Exactly.
That would make our food system way more sustainable and resilient.
Right.
That's a really strong argument.
Yeah, and it goes beyond just disease resistance.
Oh, really?
We could enhance beneficial traits,
Uh-huh.
make crops more nutritious,
Wow.
or better adapted to specific environments.
That's amazing.
Imagine crops that can thrive in droughts
Right.
or need fewer resources.
Yeah.
We could tackle some of the biggest challenges in food production.
It sounds like science fiction, but it's actually happening.
It is. It's pretty incredible.
You mentioned CLVR has already been tested in plants.
Yeah.
Can you tell us more about that research?
Absolutely.
Yeah.
There was this landmark study with a model plant called Arabidopsis thaliana.
Arabidopsis thaliana. Okay.
It's often used in plant research.
Gotcha.
Scientists targeted a gene called YKT61.
YKT61.
It's essential for the plant's survival.
Okay.
And the results were amazing.
Great.
The CLVR system spread so efficiently. Almost 100% inheritance in some cases.
That's a crazy high success rate.
It is.
But did they run into any of those resistance issues?
You know where plants might find ways to avoid the gene drive?
Right.
They did actually see some cases where plants escaped the gene drive.
Oh, so it's not foolproof.
Not completely,
Right.
but it wasn't what you might think.
Mm.
Most of these escapees had mutations in that target gene, YKT61,
Oh, interesting.
which means the CLVR system had successfully cut the gene.
Okay.
They just survived because of something called maternal carryover rescue.
Maternal carryover rescue? What's that?
It's pretty cool, actually. The mother plant provides enough of the essential YKT61 protein to her offspring,
Uh-huh.
even the ones that didn't inherit the CLVR gene drive.
So it's like a safety net.
Yeah, it gives them a temporary break.
Hmm.
They can survive even without the gene.
But they're still vulnerable to the gene drive in future generations.
Right, exactly.
So it's not a total escape, just a temporary workaround.
Yeah, nature's pretty good at adapting.
Even to our best genetic tricks.
It's true.
This brings up a good point, though.
Yeah.
If we're talking about engineering whole populations,
Right.
how do we even predict those complex interactions and adaptations?
That's one of the biggest challenges in gene drive research.
I bet.
Understanding and predicting those ecological ripple effects.
It's not just about targeting one gene.
No, it's about how that change will affect the whole ecosystem.
Right, like other species and the environment.
Exactly.
That sounds super complicated.
It is a massive puzzle.
So how are scientists even approaching this?
They're building really advanced models,
Okay.
that consider tons of variables:
Like what?
genetics, population dynamics,
Mm.
species interactions, environmental conditions.
Wow.
These models help them predict what might happen
Okay.
and identify potential risks before releasing a gene drive into the wild.
So they're like simulating an entire ecosystem in a computer.
That's a good way to put it.
That must be incredibly difficult.
It is a huge undertaking.
But it makes sense the stakes are so high.
We're talking about altering the course of evolution.
That's a big deal.
It is. The potential benefits of gene drive technology are enormous,
Yeah.
but so are the potential risks.
Right.
That's why we need rigorous research, comprehensive modeling, and open discussion about the ethics.
I agree with all of that.
It's essential.
So we've talked about CLVR in plants
Yeah.
and its potential to change agriculture. But let's go back to those animal applications for a minute.
Okay.
Specifically, the research on using CLVR to control invasive species like gray squirrels.
Yes, the gray squirrel problem.
Right.
They're a classic example of how invasive species can mess up ecosystems.
And CLVR could be a solution.
It could be combined with other gene drive strategies.
It's like what?
Researchers are looking at something called HDClvR.
HDCLVR, what does that stand for?
It stands for Homing, Daisy Field, and Cleave and Rescue.
Okay, that's a mouthful.
It is. We already know about CLVR and the daisy field concept,
Right.
but the homing part is new.
What does that do?
It's like a genetic GPS for the gene drive.
A GPS?
It guides it to its target.
Interesting.
It works by cutting the target gene
Mm-hmm.
and then using the cell's own repair mechanisms
Okay.
to copy the gene drive onto the chromosome.
Oh, so it makes sure it gets passed down.
Exactly. It's very efficient at spreading through a population.
It's like a genetic copycat.
That's a good way to think about it.
But why do we need all three components, homing, daisy field, and CLVR?
Why not just use one or two?
Yeah.
It's about creating a system that's finely tuned and controllable.
Okay.
The homing drive ensures rapid spread.
Mm-hmm.
The daisy field provides that crucial off switch for self-limitation.
Right.
And CLVR helps to minimize resistance.
So it makes the whole system more robust.
Exactly.
It's like a genetic dream team,
It is.
each member with its own special skills.
Right.
But how do we know this dream team will actually work in the real world?
You know, with wild squirrel populations?
Right. That's a different story than the lab.
It is. But researchers have been using computer models
Okay.
to simulate how HDClvR would work in a real-world scenario.
And what have they found?
The initial results are promising.
That's good.
Their models suggest that HDClvR could suppress gray squirrel populations, even in large areas,
Wow.
while minimizing the risk to other species.
That's encouraging, but I imagine there are still challenges
There are.
in actually making this happen in the real world.
Right?
How do you even release these genetically modified squirrels?
That's a great question.
And where do you start?
One approach is to carefully choose release sites
Okay.
based on things like population density,
Uh-huh.
habitat suitability,
Right.
and how connected they are to other squirrel populations.
I see.
The idea is to create a moving front of the gene drive.
Oh, like a wave.
Yeah, exactly, spreading it through the target area.
It's like a strategic invasion, but for a good cause.
That's a good way to put it.
But there must be some hurdles with this moving front approach.
One of the big ones is making sure
Yeah.
that the HDClvR squirrels mate with wild squirrels.
Right.
That's how the gene drive spreads.
So you need them to interact.
Exactly. Researchers are using spatial models to figure out the best places and times to release them.
To maximize the chances of success.
Right, and minimize unintended spread.
It's amazing how much planning goes into this.
It's not just about setting them free and hoping for the best.
It's way more complex than that.
It requires a deep understanding of the squirrels, the ecosystem, and the gene drive itself.
So it's a team effort.
It involves geneticists, ecologists, modelers, and ethicists,
Wow.
all working together to make sure it's done responsibly.
Okay, so we've got the strategy for release.
Right.
But let's get into the specifics of the HDClvR system.
Sure.
It uses multiple gRNAs, those genetic guides that direct CRISPR.
Yes, in both the homing and CLVR components.
Why is that?
Remember those resistance issues we talked about?
Yeah?
Using multiple gRNAs that target different parts of the same gene
Uh-huh.
is like having multiple attack strategies.
Okay.
It makes it much harder for resistance to develop.
Because even if one fails, the others can still work.
Exactly. It's like having a backup plan or several backup plans.
Always good to have a backup plan,
Right.
But let's not forget that HDClvR is designed to be self-limiting.
Right?
How does that daisy field component work in this system?
It's really clever, those scattered gRNAs we talked about.
Yeah.
They target non-essential genes in the squirrel,
Okay.
which means they won't cause any harm.
Gotcha.
But here's the thing. As the HDClvR spreads,
Uh-huh.
these daisy field gRNAs get diluted,
Mm-hmm.
and eventually they stop the spread.
Wait, so the same thing that helps it spread also makes it self-limiting.
That's right. It's a balancing act.
It seems counterintuitive.
It is. But at first, when the HDClvR is released,
Okay.
the daisy field gRNAs are abundant.
Uh-huh.
So the gene drive spreads quickly.
What the
The
hell?
gene drive becomes more common. The daisy field gRNAs start to disappear.
And that slows it down.
Yeah.
Exactly. It's like a self-regulating system.
That's amazing.
It ensures controlled and localized impact.
I'm really impressed with this approach.
It's pretty elegant.
It's like a genetic symphony,
It is.
with each component playing its part perfectly.
And just like a symphony, the conductor is important.
The conductor.
In this case, it's the supplementation strategy.
Okay.
By controlling when and where they release HDClvR squirrels,
Uh-huh.
scientists can fine-tune how the gene drive spreads
I see.
and ensure it's effective while minimizing any unintended consequences.
So it's a collaboration between nature and humans,
It is?
guided by careful planning and scientific expertise.
Exactly.
But all this talk about manipulating genes and controlling populations,
Yeah,
it raises some big ethical questions.
it does.
Do we really have the right to tinker with life like this?
That's a core issue. The ethical considerations are complex.
I can imagine.
We need to weigh the benefits against the risks,
Mm.
have open and transparent discussions with everyone involved,
Right. And make sure any decisions are made responsibly.
and with respect for the environment.
Absolutely.
It's a heavy responsibility.
It is, but we can't ignore it.
We're talking about potentially changing the course of evolution.
We are. And that's not something to be taken lightly.
No, it's not. So we need strong ethical frameworks,
I do.
along with rigorous scientific research
Yeah.
and open communication.
Absolutely. We need to be cautious,
Yeah,
humble, and respectful of the interconnectedness of all living things.
that's a really powerful message.
It's an important one.
And something to keep in mind as we explore gene drive technology.
Definitely. But we're still in the early stages of understanding this tool.
Right.
There's still a lot we don't know.
Like what?
Well, one area that needs more research is the potential for unintended ecological consequences.
We've talked about that.
We have, but we need even better models
Okay.
to help us predict how a gene drive might affect the whole ecosystem.
Not just the target species.
Right.
So those self-limiting mechanisms like the daisy field approach are still crucial.
We need to be able to control the spread of gene drives
Yeah,
and even reverse their effects if necessary.
just in case.
Exactly. Research into those self-limiting strategies is essential.
It's like a safety net.
It is.
But what about the public?
What do they think about all this?
Yeah, it's a pretty radical concept.
It is.
I imagine some people might be concerned.
You're right. Public engagement is crucial.
Okay.
We need to be open and honest with people,
Yeah.
address their concerns,
Mhm.
and explain the science clearly.
So they can understand.
Exactly. Transparency, trust, and education are key.
To making sure this technology is used responsibly.
That's right.
So it's not just about the science.
It's about society, too.
We need to make sure everyone has a say.
Absolutely. These decisions shouldn't be made in isolation.
Right.
We need to talk to different communities,
Mm-hmm.
respect everyone's perspective,
Yeah.
and work together to ensure this technology benefits everyone.
That's a big task,
It is.
but an important one.
It is. The potential of gene drive technology is immense,
We've seen that.
but so is the responsibility.
You can't forget them.
We can't, it's a journey we're all on together,
I think that.
and it's up to all of us to navigate it wisely and carefully.
With a shared commitment to a better future.
Exactly.
Okay, so we've talked about CLVR in plants and for invasive species,
Right.
but there's another really interesting application
Oh, yeah.
using gene drive technology to address some of the biggest challenges facing the planet.
Now that's where things get really exciting.
I'm ready.
We're talking food security disease and even climate change.
Wow.
We'll explore how gene drive technology could help us feed a growing population,
Uh-huh.
fight diseases that threaten humans and animals,
Yeah.
and even lessen the impact of climate change on ecosystems.
It sounds like gene drive technology could be a powerful tool for good.
It could be if we use it wisely.
That's the key.
It is.
Okay, so we've talked about CLVR for agriculture and invasive species, but what about disease?
Yeah, disease control?
Could this be like the most powerful application of CLVR?
It could be.
I mean, we touched on malaria before.
Right.
But could CLVR actually help us get rid of diseases that have been around for centuries?
It's a big goal. But it might be possible.
It would be incredible.
Imagine a world without malaria, dengue fever, Zika.
Wow.
CLVR could help us control the mosquitoes that spread those diseases.
Right, by targeting their population.
Exactly. We could potentially stop the transmission.
And eliminate those diseases for good.
That's the idea. It would save so many lives.
And reduce the strain on healthcare systems worldwide.
Absolutely.
That's a future we can all get behind.
I think so.
But wouldn't getting rid of all mosquitoes have unintended consequences?
You mean for the ecosystem?
Yeah, they are part of the food chain.
That's a valid concern, and scientists are working on ways to make gene drives more specific.
Oh, yeah.
So they can target only certain species or populations of mosquitoes.
The ones that are most dangerous to humans.
Right, like they're looking into using CLVR to suppress only the mosquitoes that carry and transmit malaria.
So that would minimize the impact on the rest of the ecosystem.
Exactly. It's all about finding that balance.
Using the power of gene drive without causing too much disruption.
Right.
But even with those safeguards in place, manipulating entire species still feels ethically complicated.
Whether it's mosquitoes or squirrels.
Yeah, it raises big questions.
It does. The ethical considerations around gene drive technology are huge.
I can see that.
They're talking about potentially changing the course of evolution.
It's a lot of power.
It is, but we have to be careful, transparent, and humble.
So it's not just about the science.
It's not.
It's about our values, our relationship with nature.
And the future we want to build.
It's a conversation everyone needs to be a part of.
I agree. Scientists, policymakers, and the public.
So where does that leave us? We've explored CLVR from its molecular mechanisms to its potential applications.
In agriculture, conservation, disease control.
We've talked about the ethical challenges and the need for responsible use.
It's been a journey.
It has.
Yeah.
We've seen how this technology could change the world.
For better or worse.
But it's also made us think about our role in shaping the future.
And our responsibility to all living things.
It's definitely left me with more questions than answers.
That's okay. Asking questions is how we learn.
And it's how we make sure we're using this technology wisely.
You got it.
Well, I think that's a great place to wrap things up.
I agree.
I hope this deep dive has sparked your curiosity.
And got you thinking about the future.
There's so much more to learn about this topic.
And we encourage you to keep exploring.
So until next time, keep asking questions, keep learning, and keep diving deep.
We'll see you then.